welcome. It's a great pleasure for me to introduce Katia Koelle. She's a professor of biology at Emory University and received her PhD in ecology and evolutionary biology from the University of Michigan. She was then a postdoc at the Penn State Center for Fluid Dynamics with Brian Grenfell. And yeah, she has done a lot of great work. I think this is all. Oh, they put this on. Yeah, she has done a lot of great work on developing medical models to improve our understanding on RNA virus evolution and disease dynamics at various scales, like both at the within host and also between host level, but also on a population level scale. And she applies the models to various pathogens, including influenza, SARS-CoV-2, and norovirus. Great. Good to have you. And you take it away. Thank you, Brian. All right, can everyone hear me? Yeah, great. Well, thank you all for coming. It's great to talk to a lot of you, throughout the day, and it's great to see some familiar faces. So again, yeah, thank you so much for coming. What I'm going to talk about today are really viral transmission bottlenecks, and I'm going to weave that in with some work that we've been doing on looking at patterns of disease severity and how estimating transmission bottleneck size actually helps us understand variation in disease severity or perhaps not. Okay. Usually, should these buttons and Texas.
I just used the mouse to click on it.
Oh, yeah. Can I go back?
Try.
Arrows don't work. Oh, great. Great, fantastic. You should be 19, I guess. Okay. All right. I know a lot of you actually think about transmission bottlenecks, but just to get everyone on the same page, what I wanted to do first is just introduce what they are, right? And so we have a situation of a transmission event, right? And we've got, people see this pointer, right? And we have a donor individual and we have a recipient individual, right? And the donor individual is infected. They might be infected with a lot of viruses or kind of viral particles. Let's say if they're infected with flu or if they're infected with SARS-CoV-2. Right? And during this transmission event, what happens is that some of these viral particles get expelled from that donor, and those particles, some of those particles might land in the respiratory tract of the recipient. Right, and then some of those, a subset of those, will be able to kind of infect cells and then establish their own kind of genetic lineages in the recipient host. Right, and so what transmission bottlenecks. Sorry. Okay, thank you. Okay, so what transmission bottlenecks is depicted here. This is the number of surviving individuals really that stem from the donor population that go on to establish a genetic lineage in this newly infected individual. This is also known, transmission bottlenecks are also known as population bottlenecks. What I wanted to clarify is what transmission bottlenecks are not. So it's not the number of virions or viral particles that land on the recipient. That could be a much larger number, with only a subset actually establishing infection. And it's also not the number of unique viral genotypes that establish infection. You can see, for example, here there might be, the bottleneck size might be something like 12, but only 2 genotypes, this yellow and the blue genotype, actually go on to establish. So the genetic bottleneck is 2, but the transmission bottleneck size or population bottleneck size is 12. Okay. So my group has been very interested in actually quantifying transmission bottlenecks over the years. We've developed some methods, and I'll talk about those for a little bit today. But we're interested actually in quantifying these bottlenecks for several different reasons.
Are they still saying it's echoey?
You're unmuted.
It's going, okay. So we're interested in quantifying these transmission bottlenecks for several different reasons. One is, I know many of you are also very interested in trying to infer who infected whom in an outbreak setting, right? And if transmission bottleneck sizes are large, you can imagine that minor genetic variation that evolves within a single infected individual could then be transmitted, right? And new minor genetic variation can also evolve, right? And so if you have large transmission bottlenecks, you have the hope of actually reconstructing from an outbreak setting who infected whom by looking at that shared genetic variation, shared minor genetic variation. If transmission bottleneck sizes are very small, right, minor genetic variants are generally not transmitted, right, and then it becomes very difficult, if not impossible, to actually infer who infected whom. So very practical reason, for wanting to estimate transmission bottleneck sizes. Second reason is actually just basic evolutionary understanding of constraints to viral adaptation or pathogen adaptation more generally. So generally, it's thought that if a donor is infected, right, and there's a beneficial mutation that might arise in that donor, the frequency of that allele might increase, right? And if the transmission bottleneck size is sufficiently large, that beneficial variant can be passed on and could, again, kind of increase the frequency in that recipient individual. So when transmission bottleneck sizes are large, pathogen adaptation at the population level might occur pretty rapidly, right? But when transmission bottleneck sizes are small, on the other hand, beneficial mutations such as this might not be transmitted, right? And so it's thought that population level pathogen adaptation can be slower when bottleneck sizes are smaller. There's a lot of really interesting work by Santi Elena and Mark Zwart, which actually kind of argue that small bottleneck sizes, for example, there's some benefits to the viral population, such as being able to cross crops and fitness landscapes and so forth, and purge themselves of deleterious cheaters. But this year, our understanding of the factors that shape and the speed of viral adaptation should be impacted by transmission bottleneck sizes. And what I'll really focus on today is this third point, which is that understanding transmission bottleneck sizes might actually help us gauge the potential impact of control measures. So let me go into this into a little bit more detail. If you think back to 2020, if you haven't repressed that year, there were a few very high-profile opinion pieces or perspective pieces that argued the following. So this was before the COVID vaccines rolled out on the market, right? And the idea behind these opinion pieces was that an individual who wore a mask, right, they would protect themselves from infection, right, because masks lower the probability of getting infected. And if an individual is infected wearing masks, they might actually have lower transmission rate. Right? But beyond that impact of masks, these articles were arguing that if you wear a mask, if you happen to get infected still, right, so there's a breakthrough infection, the number of viral particles or the knockdown dose that you receive in your respiratory tract might be lower than it would have been otherwise, because the mask might keep out some of those viral particles. Right? And so the idea was that with the lower knockdown dose, your risk of developing severe disease might actually be lower than if you had a large knockdown dose. Right? So that was the idea, and this was coined the variolation hypothesis. It's called the variolation hypothesis because of a practice that was started in, I believe, about 1600 or so in East Asia. It's called variolation. There's a response to smallpox, variola, right? And smallpox had about a mortality rate of about 1 out of 3, every 3% mortality rate. But individuals who survived a smallpox infection were then permanently immune to reinfection. So the practice of variolation started where individuals who had a previous smallpox infection recovered from that infection. Oftentimes they had these scabs, smallpox scabs, and those scabs were taken and then pulverized. And then the practice was that those pulverized scabs were basically basically shot up the nose of someone who was naive, so had not been previously infected. And that individual had only about a 3% chance of dying from that practice. But about 97% of the time, they basically developed an immune response, right, from those pulverized scabs, and were then no longer susceptible to the naturally circulating smallpox virus. So this practice, which had a 3% mortality rate, was improved upon over the generations, and so it turns out that mortality rate from the inoculation was actually reduced by inoculating on the hand rather than directly up the nose, and so forth. The reason why this is called the variolation hypothesis is presumably because with these pulverized scabs, potentially kind of the number of live viruses that were in that powder might have actually been lower than one would have received with a natural infection. This argument, I haven't actually found a lot of documentation of that in the literature, but that's why this is called the variolation hypothesis. The lowering the inoculum dose might actually produce lower disease severity or lower mortality. Okay, so that's the hypothesis. So what I'd like to do today is look in more detail at that hypothesis and then bring in kind of estimates of transmission bottleneck size, which are related to inoculum dose. So what we're going to do in the next 30 minutes or so is I want to guide you through kind of these two predictions that we can do get to using modeling approaches and also data analysis. So what we're going to do is look at the effect of inoculum dose, which is related to the transmission bottleneck size, right? We're going to look at the impact of inoculum dose on the risk of infection, right? So at a higher inoculum dose, presumably your probability of infection decreases, right? But what does that relationship look like? And then here I want to look at the relationship between inoculum dose and the risk of developing severe disease, which is some quantity of quantification of disease severity. Okay. And then what I want to do is use the viral sequence data to tell us basically where we stand in terms of inoculum dose in natural infections. Okay. And this is really to kind of evaluate the plausibility of this variolation hypothesis. Okay, so for that first graph that we want to populate, what we can do is we can use a branching process model. So many of you are probably familiar with branching process models. These are stochastic models which are parameterized with an offspring distribution. And we're going to use this branching process model to figure out the relationship between inoculum dose and infection risk. So offspring distributions can be generally specified. So here I'm showing just an offspring distribution, which is a Poisson distribution characterized by a mean number of offspring, here 15. And here I'm showing an alternative offspring distribution, which is a negative binomial distribution with some very high level of overdispersion. So the mean here is 2.6 offspring, but many individuals have 0 offspring, and there's fewer that have, very many. But the mean is 2.6. And what we can do with one of these branching process models, once we've actually parameterized the offspring distribution, we can forward simulate it, right? And you can see whether, starting here with one single individual, right? You can see whether that lineage goes extinct, as you see here, or ultimately establishes in the population. It's a super critical process because we're going to consider offspring distributions that have kind of a mean greater than 1. Okay? So we can also, instead of just forward simulating, we can actually do that analytical calculations based on these offspring distributions and come up with an analytical solution to what the probability is of one of these lineages establishing. Okay? So we're going to do this in the context of actually thinking about individual infections, right? So the individuals in this branching process models are believed those infected cells. So we think operationally we can define the inoculum dose as the number of infected, initial number of infected cells. Okay? So then we have our initial population size, and we can ask whether those infected cells that you have initially, whether it goes on to kind of produce a successful infection, right? So whether that infection establishes. And so what offspring distribution should we use? What we can do is we don't actually know the correct offspring distribution, right? But we can provide kind of the model with some reasonable range of offspring distributions. And again, this is the offspring distribution we're thinking about is from a single infected cell. How many infected cells do they leave in the body before that infected cell dies? Okay. So what we did was we looked at a very broad range of offspring distributions. Here, as different rows, we have different values of within host R naught, which we got from this Ruud Key and Harrelson paper that was published. And so by fitting within host models to viral titer data, they estimated that the within host R naught of SARS-CoV-2 was between 2.6 and 14.9, and on average it was a 7.4. And so what we decided to look at are this minimum value, that average value, maximum value for R naught. This within host R naught is also kind of the mean of the offspring distribution. And then here across these columns, we can consider a broad range of overdispersion. So this is kind of cellular heterogeneity. So if all cells behave very similarly, we have a very short coefficient of variation here, right? Not the distribution like one of these, right? But there's a lot of cellular heterogeneity with some cells some infected cells producing zero viral progeny, whereas a small proportion produce a lot of viral progeny. That would be more consistent with one of these highly overdispersed models. Okay, and we know from some microfluidic work of other groups, and as well as some work from Jesse Bloom's group that there's actually considerable cellular heterogeneity in at least in influenza, and we would expect it probably also to be the case for SARS-CoV-2. So we're going to look across these very broad ranges of parameter space because we don't know where we actually lie. So once we've done that, right, we can plot up kind of our analytical results here. So what I'm showing here is within a given color, these are the 3 different R naught values, right? And so if we're given an inoculum dose of, let's say, 100 initially infected cells, with a low R naught, your probability of infection might be here, 55%. With a higher R naught, it might be, 62%. With an even higher R naught, it might be 67% or something like that, right? So the higher the within host R naught, the higher the probability of infection given a certain inoculum dose. Given a certain inoculum dose, also the higher the level of cellular heterogeneity, the lower the probability of infection. This is very similar, for example, to the super spreading paper that some of you are probably familiar with, Jamie Boyd-Smith, where the more transmission heterogeneity there is, the population level, right, the lower the chance of actually the pandemic being kind of established. So this is the same exact results, kind of only at the within host level. And so you can see that higher levels of cellular heterogeneity for a given R naught reduces your probability of infection or increases the probability of infection going extinct. Okay. What I wanted to point out, though, was even at these extreme high levels of cellular heterogeneity and low R naught, when you get to about 1,000 infected cells initially, you're pretty sure that you're going to be infected, right? So this goes up to 1, between here 10 to the 3rd, 10 to the 4th, right? And this is really at the extreme level. It seems like we're probably more likely kind of in this area of kind of parameter space in terms of these this level of cellular heterogeneity. So when the inoculum dose is, let's say, under 1,000 or under 10,000 initially infected cells, you're almost surely going to become infected, right? But reduction in the inoculum dose in this range, by 1 or 2 orders of magnitude, should actually decrease the probability of that you get infected. Okay. Okay. So what I'm going to do next is transition over to this plot over here. And try to populate that given work that's been done in the literature. So here, I know that many of you work on within host models. So here we also decide to kind of make use of within host model for acutely infecting virus. A lot of these models have been developed in the past for influenza virus, both by Alan Perelson's group and then also Brian Grenfell's group. These models generally look something like this. We have uninfected cells. These are compartmental models within hosts. So uninfected cells, these uninfected cells become infected by free virus entering them. Infected cells can be have this background mortality rate and can also be killed by T cells. They release interferon. Interferons decay. Interferons also have the impact of they also kind of make cells refractory to infection if they contact uninfected cells. And these refractory cells are like recovered cells which have never become infected, and they can't become infected themselves. And of course, infected cells produce free virus. This is taken from a paper that was published in 2021 in PNAS by Ruben Keegan, Alan Perelson again, where they used viral titer data. I believe it was from the NBA data set. Jonathan Graham might have shared it with it, I'm not sure. So here we have the data, and what they did was they fit this or a very similar model to this to those data to estimate parameters of this model. They set some of the parameters and they estimated some other ones. And what we did was, in addition to kind of using these equations, we kind of tacked on an additional equation based off of previous work that Bustamante had done, which was looking at patterns of tissue damage during an infection. So this equation that we tagged on, tissue damage increased based off of interferon levels and the T cell killing of infected cells. And then overall disease severity, we can think of this just kind of integrating over kind of the tissue dynamic, tissue damage. Okay, so that's how we assess tissue damage. Okay, so what you're seeing here is actually some viral dynamics from that within host model, parameterized with kind of the mean parameters from the Key and Perelson paper. Here it's for 3 different inoculum doses, yellow, orange, and red for very high inoculum doses. And you can see here, first of all, for the low inoculum dose, so these are 10 initially infected cells. What you see is, this is days post-infection, and you see kind of these viral dynamics, and you see virus kind of exponentially growing, and then declining and resolving within about 15 days or so post-infection. If you look at the viral dynamics here for a higher inoculum dose, a considerably higher inoculum dose, what you see is very similar dynamics. So you see here, only they happen a little bit earlier. So the argument here would be that if you get a larger inoculum dose, right, if you get infected, you might develop, potentially symptoms, or your peak time to peak viral titer might be shorter than if you had gotten a lower inoculum dose. But if you look at both of these dynamics, if you look at the tissue damage dynamics that are associated with these viral titers and interferon dynamics from over here, you can see that tissue damage also looks very similar, except for with the higher inoculum dose, that tissue damage happens earlier. But overall, disease severity and tissue damage is very similar between these 2 inoculum doses. However, if you go to a much higher dose, right, again, that makes the infection earlier, but the peak is also much higher. And you can see that tissue damage is all earlier as well as kind of much higher, and you would expect greater disease severity as a result. Okay, so this is using within a host model under the same under one parameterization, right? With the initial conditions just being different between these 3 different cases. Okay, so instead of just doing it for 3 different inoculum doses, what we're going to do is just look at a lot of different inoculum doses under that parameterization, and you see that here in the solid black line. Okay, disease severity again being kind of the summary of these kind of tissue damage dynamics, just the info under that curve. What you can see here with this exercise, right, is that disease severity stays, constant, so does not depend on inoculum dose until inoculum dose is much, much higher, right? Is close to kind of, 10% of the target cells that you actually have for SARS-CoV-2. What we then also did was we looked at their range of parameter estimates, and we kind of sampled from those parameter estimates to actually see kind of what we would expect under different parameterizations. And under different parameterizations, it's very similar. Disease severity doesn't change until the very end, with very high inoculum doses. And at that point, disease severity increases. So I want to point out here that, it's for each one of these, it's relatively flat until the end, right? But you can also see a huge amount of variation in disease severity, right? At any given inoculum dose, right? And this results from kind of those underlying different parameterizations. Those parameterizations differ in terms of how strong your innate immune response is, so host factor, how quickly your T cell responses, and also the viral replication rates or data kind of at the within host model, which could reflect viral factors such as strain differences, right? Which might impact disease severity, right? Delta versus Omicron, for example. So there is so this variation here at any inoculum dose is really due to differences in host factors and differences in viral counters. Right? The differences in inoculum dose don't impact the predicted disease severity, unless the inoculum dose is extremely high. This shows exactly, this plot shows exactly this, except for it's just normalized by the disease severity when there's 10 infected cells, just to more clearly show that that full change in kind of disease severity, there's no difference really until you get to much higher inoculum doses. Okay, so we got these two plots here, right? And so what you can see here is that the sensitivity of the risk of infection to inoculum dose, there is a relationship there, right? But that it's really kind of at these low inoculum doses, whereas disease severity, right, also depends on inoculum dose, but it's really only at these very, very high inoculum doses. So then the question is, well, where are our natural inoculum doses, right? Because if the inoculum doses are around here, then reducing an inoculum dose through something like masking would reduce your probability of infection, right? But it would not have any impact really on disease severity. Right? But if the natural inoculum doses were actually kind of up here, then wearing a mask should not really impact your probability of getting infected. You're likely to get infected anyhow with the inoculum dose inside, right? But they would impact disease severity.
Okay. Yeah. So vaccination and infection, are those, do you consider those as shifting those curves rightward for the likelihood of infection given an inoculum dose? Or how do you figure those work?
And that's a really great question.
If that's sorry, the second part of that question is if that's how that works, it's hard to understand how that would work in terms of inoculum dose relating to severity of infection. It would suggest that it has to be through some other mechanism, right? Because that to keep it with what you're seeing in the right graph here.
Yes, that's a really, really great question. Maybe we can. Do you want me to talk about that at the very end?
Yeah.
It's a fantastic question, and I think we should probably spend some time on it.
Okay.
Yeah, so whether do we think of vaccination as kind of modifying? So we think of masks as modifying the inoculum dose, right? But whether we can think about vaccination as modifying an inoculum dose or having some other impact, we definitely should discuss after. Yeah, it's a great question. Okay, so this gets us to kind of this question of, okay, well, we have these different predictions for under different inoculum doses, right? So where are natural inoculum doses? Are they very high or are they actually very low? And so this gets us to kind of transmission bottleneck sizes, which are related, but not identical to knockdown doses. But we can actually mathematically convert knockdown dose into transmission bottleneck size relatively easily. There's some modeling assumptions. But basically how transmission bottleneck sizes are currently being estimated. So we've done a lot of work in this area over the years. There's a few different approaches, right? So if we have a donor individual and we take a sample from that donor and deep sequence it and then call variants, right? We can do the same for a recipient that we know received the infection from this donor. There's a variety of different approaches in the literature. The closest one is known as the presence-absence method. So you just look at the genetic variation that's present in the donor, and you say, how much of it is transmitted, right? And the less it's transmitted, the lower the transmission bottleneck. Right? And you can kind of estimate it quantitatively. The other way to do it is using kind of just binomial samplings, right? Fisher model. And which is, I think, an improvement over the presence-absence method. What we've developed by now, 6 years ago, is what's known as the beta binomial approach. So it takes into consideration kind of the stochastic viral growth, in the beginning of the infection, especially when transmission bottleneck sizes are small, we have to consider that stochastic growth. Basically by taking into consideration the frequencies of these isomers in the donor and the frequencies of isomers in the recipient, we can estimate this family population size of the transmission bottleneck size. There's a haplotype based extension of this beta binomial approach that Chris Illingworth and Montefari have developed. But all of these existing approaches really rely on the subset of loci in the donor that are polymorphic, right? So you look at kind of how much of the genetic variation you're seeing here is effectively transmitted. And this approach has been used both, on flu. So this is, I think, really nice work by Adam Warren's group and J.G. Crone leading it, where they plotted here the frequency of an iSNP in the donor and the frequency of that iSNP or intrahost signaling variant then in the recipient. And what they see just visually in this plot, right, is that iSNP that's present in a donor, even at high frequencies, is rarely seen as kind of polymorphic in the recipient. It's either kind of fixed or it's lost, right? Which indicates that there's a very small transmission bottleneck. And they've also applied that beta binomial approach and other approaches to estimate the transmission bottleneck size on the order of 1 or 2 viral genomes. We've used that our beta binomial approach for SARS-CoV-2, where we estimate similarly that the transmission bottleneck size is on the order of about 1 to 3 viral genomes. So very small. And of course, for SARS-CoV-2, tons of people have done analyses such as this. I'm using a variety of different approaches and a variety of different data sets. They're all very consistent with one another. So this paper by Katia Koelle also finds very small transmission bottleneck sizes. This paper that I was fortunate to kind of collaborate on with Louise Mothla and Tom Friedrich and Kat Braun also found very tight transmission bottlenecks. You can also look this in the experimental transmission studies, and you also find very small transmission bottlenecks. So this is all well and good. But then kind of I had this nagging thought in my head over the last year or two, which kind of arose from the following kind of arguments, and really kind of point towards some of these methods actually having the potential to vastly underestimate true transmission bottleneck sizes. So the first thing that we should kind of consider is that the sample that you have from the donor is probably not exactly at the time of transmission. It's probably within 1 or 2 or 3 days of the transmission event. And so if you look at this analysis by Crone and Adam Warren's group, what they looked at was longitudinally within acute infection, right? Across these individuals for which they have longitudinal data from the Hive study in Michigan. They called iSNPs, on each one of the sample days, right? And what they're plotting here is the time between those within host samples. Right? They color code them depending on whether that iSNP was actually present in both samples, only in the first sample or only in the second sample. the first thing you should see is that there are a lot of red and yellow dots here, meaning that if an isomib is detectable. Still hear me? Yes? Okay. If an isomib is detected on one day, oftentimes it's not detected on the next day. It might be detected at around 20% frequency on one day, the next day you don't find it, right? Or it's not detected on the first day, and then you see it on the next day at 15% frequency. So that happens often, right? And then when you do find it present in both samples, even within like a day apart, sometimes there's a huge change in real frequency. So this means that if the time that you're sampling from the donor and the time of transmission is actually off, even by like a day, right? The impact that this would have on estimating the transmission bottleneck size would be that you'd systematically underestimate the transmission bottleneck size, right? And that's because, let's say, you can imagine there's a very large transmission bottleneck size, so you would expect the recipient isomers to mirror the donor isomers, right? But if there's a very dramatic change in isomer frequencies from one day to the next in the donor, right, that's going to kind of reduce the kind of correlation between isomer frequency between the donor and the recipient, and therefore kind of decrease the transmission bottleneck size that's estimated. Yeah.
So sort of strong selection with something that's complete like this sort of estimation completely hopeless just to due to.
Yeah, that's a really great question. So all of these approaches that I talked about here, these assume that all genetic variation is neutral. There has been an extension which has looked at basically relaxing that assumption. Yeah, but everything here is generally kind of considered neutral. Okay. So the second issue is that all of these methods also assume that there's random sampling from the donor, right? But we know from a lot of virological studies that there's viral aggregation can occur, right? And viral aggregation oftentimes occurs from viral particles that are close to one another, spatially in the respiratory tract, right? And so are much more likely to be genetically the same, right? And so because of that non-random sampling, that is also going to have the impact of actually reducing or vastly could have the potential to vastly underestimate transmission bottleneck sizes. Okay? So I'm a little worried about kind of the results, in terms of transmission bottleneck size, given kind of all these previous analyses. So what we decided to do was actually develop an alternative method that was not sensitive to these assumptions. And the approach is as follows. So we're not going to look at isomers and genetic variation that's in the donor, and we're not going to ask about how much of it is transmitted. We're actually going to look at it as kind of a very different subset of the data. And that subset are what we call clonal variants. So what are clonal variants? Clonal variants are variants that are absent from a donor and fixed in the recipient. So on one of these plots that I showed before, where we have frequency of a variant in the donor against the frequency of the variant in the recipient, these fall right here. So it might be that at site 100, the donor has G's, in all the reads, right? And the recipient has all C's, right? So if you look at this influenza A virus data set from Adam Loring's group, we can go across all the transmission pairs, and we can quantify how many clonal variants are in each one of those transmission pairs. And what we find is that 42 transmission pairs have 0 clonal variants, so there's no dot up there, right? 5 transmission pairs have 1 clonal variant, 3 transmission pairs have 2, 2 transmission pairs have 3, and 0 transmission pairs have 4 or more. Okay? So this looks like pretty low dimensional data that you're getting from kind of all of this deep sequencing data, but I think it's actually potentially more useful for estimating transmission bottleneck size. Okay, so what do we do with this data? What we want to do is we ultimately want to estimate the initial number of viral particles that are starting off infection. So the underlying model that we're going to use is again a branching process model, where we have an initial viral population size. So we have n wild type viral particles. Okay? Wild type just means that they come from the donor, right? They can be genetically different, but they all come from the donor. Okay, so to kind of parameterize this branch across small, each viral particle is going to have some offspring distribution, with the mean number of offspring being within host R naught. And then mutations occur during the birth of the viral particle, and that's just with a mutation, per genome mutation rate of mu. Okay. So this just shows, our overall kind of offspring number. This geometric distribution we're assuming. Of that, the wild type offspring are shown here in dark green, and then the mutant offspring distribution is shown here in light green. And what's nice about a geometric offspring distribution is that you can kind of partition it into two separate kind of negative binomial distributions for mutant and wild type. So that's our underlying branching process model. It's a kind of multi-type branching process model. So if you think about this model and the types of outcomes that could result from that model, they're the following. One thing that can happen, so here is a depiction with just two initial viral particles in each one of these simulations. So here we've got our two initial wild type particles, right? And we see kind of these lineages, was kind of stuttering along. And it turns out that in this case, we have a wild type lineage that establishes, right? See that here? This one establishes and this one establishes. So when there's a wild type lineage establishing, right? That means even though these mutant lineages get made and potentially kind of establish themselves, right? You're not going to see any clonal variants in this case. Second possibility is that there's this branching process model, and all of these viral particles ultimately go extinct, right? In this case, the recipient's not going to get infected, right? And we're not going to actually see any clonal variants because that's not going to be a realized transmission pattern. Okay. So here what we have, let's jump to this one. Here we have the wild type lineage going extinct, but we have, before it actually goes extinct, it seeds two or more mutant lineages, right? And both of those were more, established. In this case, if we assume, sites, it's unlikely that these mutations here that created this mutant virus occurred at the same exact site that's created this mutant virus, right? And so in this case, you also don't see any clonal variants. So the only way to get clonal variants is in this scenario, right, where you have the wild type lineage going extinct, but before it goes extinct, it seeds one successful mutant lineage, right, that then establishes. Right? And so what you have here is one or more mutations here, or you can have kind of more, and you can have more kind of clonal variants kind of establishing kind of within even this lineage, right? So this is a situation where you can get one or more clonal variants, and we can kind of resolve whether it's 1, 2, 3, 4, et cetera, clonal variants, kind of using a recursive approach. So it turns out, so these are the 4 different possible outcomes, and we can actually derive analytical expressions for the probabilities of each one of these outcomes. Yes?
Just 5 more minutes.
Okay, great.
Just do the time check.
Okay, great. Perfect. So we have kind of our analytical formulas for actually being able to say what's the probability of each one of these occurring, which means then we can actually estimate, given the data that we have from our transmission pairs, right, how many clonal variants, and how many transmission pairs have this many clonal variants. What we can do is actually estimate the initial number of particles and the mutation rate. So here, instead of just a constant number of initial particles, we just assume that's a Poisson distribution with some mean lambda, right? And what you can see here is the likelihood surface. So very small number of initial particles and mutation rates that are kind of on the order of 0.7 to 3. We can translate this actually into mean transmission bottleneck size, right? Which we also kind of here have that it's very low. This is with an R naught of 11.1 for flu. The mean NB, as I said over here, is very small. It's on the order of 1 to 2 viral particles. And the mutation rate is between 0.7 and 3.0, per genome per replication cycle, which actually agrees with some other work out of Emory's group, which used kind of this called classic fluctuation test to estimate this mutation rate. So it's consistent with data. For SARS-CoV-2, we have these are our data, and again we estimate a very, very small percentage of all size. And the per genome mutation rate is consistent with some estimates from the field borders group. Okay, so given a result like this, that bottleneck size is about 1 to 3 viral particles, we can go back to our branching cost model that looks at kind of disease severity and so forth, and we can actually convert kind of the inoculum dose to a mean bottleneck size. So we can estimate, given if we have our mean bottleneck size estimate, right, which you can see here, we can see what that corresponds to in terms of inoculum dose, right? And it corresponds to about 100 initially infected cells or less, depending on what your assumption is for cellular heterogeneity mostly. Which basically means that we are in this area of parameter space, right, with a relatively low inoculum dose, natural inoculum dose. Which means that if you think about masking again, which is acting to reduce the inoculum dose, this would indicate, if you just look at one of these lines, right, that if you're reducing the inoculum dose by wearing a mask, the impact that would have in terms of your probability of getting infected, that would decrease your probability of getting infected, right? Which makes sense, that's been kind of established in many studies. However, here at these very low inoculum doses, a decrease in the inoculum dose would not actually reduce disease severity. Okay, so we think that the variolation hypothesis is theoretically implausible, based off of these modeling analyses. What we also wanted to do though was actually go into the experimental literature and look at experimental challenge studies and experimental transmission studies to see whether there's actually any support for this variolation hypothesis. And I know I don't have time to kind of go through these, but there's very little support for the variolation hypothesis. So studies like this, for example, individuals for Syrian hamster study, I think. Contact animals were placed with donor animals for different amounts of time during different peak viral loads and so forth, right? And some hamsters got severely sick, some didn't, but there was actually no correlation with kind of how long they spent the donor or at what point in their viral cycle. In terms of studying disease severity. This one I thought was really interesting. It turns out that they found that masking hamsters between cages actually reduced the probability of recipient hamsters getting infected. Right? They also concluded that it reduced average disease severity, but they didn't condition on infection. So effectively, the ones that didn't get infected when masked had a 0 severity score. So if you remove the ones who didn't get infected and do the analysis again, the analysis is really significant. And there's some other interesting things, but I'm going to find out. And yeah, there seems to be very little empirical support for the variolation hypothesis. In the original studies, in the original prospective pieces, the argument would be something like on these different cruise ships, there were different rates of symptomatic disease, and on this cruise ship with lower symptomatic disease, they wore masks, and on this one with more symptomatic disease, they did not wear masks. And I tried as hard as I could to get the age distribution of people who are on the Princess Diamond versus going to Antarctica on a cruise ship. And I could not. I got the Princess Diamond once, but I could not find the age distribution of people who went to Antarctica. But there's a lot of confounding factors there. So I thought, going to the experimental challenge and transmission studies that would be more controlled situations, scenarios. Okay, so in conclusion, bottleneck sizes seem to be very small for SARS-CoV-2 and seasonal influenza viruses. As such, NPIs that lower inoculum doses, masks, improved ventilation, et cetera, can decrease the probability of infection and possibly increase the duration of time from infection to symptom onset. Right? Or increase in the incubation period. However, it seems that disease severity is largely insensitive to inoculum dose, except for at very, very high doses. So those NPIs are unlikely to reduce the probability of developing severe disease conditional on infection. So I think this variation hypothesis came back kind of from, some intuitive kind of feelings from the field, really from toxicology. I think where if you think about kind of light exposure or something like that, and more light exposure, right, the more severe your symptoms will be, right? The light is not a replicating agent, right? So I think for replicating agents like these viral agents, if you think about natural exposure levels, I don't think that variation in natural exposure levels, really have the potential to impact kind of the extent to which you get severe disease. And with that, just some acknowledgements for folks who have contributed to this. So Teresa, Jeremy, and Mike all were heavily involved with the transmission bottleneck size estimation work from the de novo genetic variation that he recently published. And then Jack, Julie, Rustam, Anise, and Daniel on the variation hypothesis analysis. That maybe I can go back to it. the question. Yeah, thank you.
All right, thank you for the great talk. Are there any questions?
So it seems like the severity of disease is very robust to all the parameters. It goes up at about 10 to the 7,
Yeah.
despite all the different changes parameters that we have. Is
Yeah.
there like a fundamental constants underlying that? Would that depend on something that is very well known? Like, can you give us some treatment
Yeah.
for that?
That's a great question. So I don't know whether there's something really fundamental about it. So once you get the way we thought about this access was really here estimates of the number of epithelial cells that are actually that are susceptible, to infection is on the order of about here 10 to the 7.5 or something. So this is already kind of very high. You can't go higher than this, basically based off target cells, right? So I don't know whether it has something to do with it being kind of the 7th or a certain percentage of kind of the total availability of target cells. But it's something definitely to look into. I mean, you can kind of see, that initial peak, right? And I think it has to do with, the speed of the effectively the T cell response, in terms of limiting viral replication kind of, early on. Yeah.
Yeah, so with the clonal variants that with the branching process, right, over time, it eventually comes down to like an exponential growth.
Yeah.
And so with the empirical dynamics, they're actually, there's a demographic
Yeah.
process. So, what effect do you think that has on your ability to estimate the number of clonal variants and whether or not you see any sort of changes depending on how long ago the infection was?
Yeah, that's
Or
what
the sample was.
Yeah. So we thought about this a lot, mostly because if you think about oops. What is that? Oh, gosh. This one. Okay, so we thought about this, and it was mostly because we didn't know whether the same problem of the sampling time, right, from the donor or from the recipient, right? And for the recipient, if you start off with a small viral population, and you have this kind of stochastic growth, obviously you're not going to get to, infinity, right? You kind of level off, and then you also have these low effective population sizes in the recipient, right? And so we were worried about kind of whether our results would be sensitive to that. So what we did was we actually looked at some longitudinal data from a bunch of infected hosts that were or were not part of transmission pairs. And we looked at how many actually over the time of sampling, how many clonal, how many variants actually kind of, effectively kind of would be kind of come clonal over the course of infection. Right. And so there's very, very few. right? So if there's a kind of clonal variant that you see in a transmission pair, right? It doesn't become, it doesn't become subclonal again, right? It doesn't go to zero again. And so I think it's pretty robust to that. There were a couple of cases actually where when the thresholds you set actually impact the number of clonal variants you get, right? And so if you have, if it's 100% exactly, you might call something a clonal variant, but if it's at 99.5%, you call that a clonal variant. If it's at 0% of the donor, right? And so there were a couple of instances where we looked at, sensitivity to that variant calling threshold, and didn't impact it much, but we just wanted to make sure because at 99.5, the next day, it could be at 100%, right? And so we were, it wasn't very sensitive to that threshold. So other thing I want to say about that. Yeah.
With the method that you presented during the similar estimate for the bottleneck size, it seems like maybe like some of these concerns were not happening in the data set, or they weren't like, can you like from there try to give guidance on like how 90 days apart you can do symbols and it's so
So I think the reason why so I think our approach if it had actually estimated large number transmission bottlenecks. Well, first of all, it loses resolution kind of at larger transmission bottlenecks. So with larger transmission bottlenecks, you actually get no clonal variants, right? And so once you're actually at zero clonal variants, your highest likelihood estimate is infinity number of viral particles, right? Initially. So it's actually really only good for when there's a small transmission bottleneck, it kind of provides resolution there. But I think it could have actually, based off of numerical simulations and simulated data sets and so forth, it could have estimated that it was 4, 6, or 10 or whatever, right? Viral particles. And other approaches would then have been underestimated. So I don't think that the agreement of those two numbers then indicates that these aren't problems, right? I think because the number is so low, even with these biases in here, it's estimated to be low. I mean, but in actuality, if it is low, right, well, these methods would actually estimate it to be low, right? Because this one can't estimate it to be effectively higher, right? But the one other thing I wanted to mention was there is a little bit of bias I think that can be introduced, in this method if the identification of transmission pairs relies on, so when people try to identify transmission pairs, right? Some say, well, it's only a transmission pair if we know that they had contact and then the symptoms are this many days apart, or at most this many days apart, and if those viruses are genetically pretty similar, right? So sometimes they put like an arbitrary cutoff right here, which would actually, with that arbitrary cutoff, and they say, if it's 5, SNPs, if they're 5 SNPs away from one another, maybe people don't think it's actually a real transmission pair when it is, right? But that would bias these estimates to be high.
Okay, I think we're
Okay.